Hello, so today I'm presenting my project characterizing transcription factor binding with multi-omics sequence model. So the linkage between transcription factor and regulatory region is a crucial step for GRN, for building GRN, and that step requires two types of information: the sequence usually used for motif matching, and expression and accessibility, or the correlation between them. So usually analyzed to give cellular context to this motif matching. And our goal of this model is to combine the two sources of information and improve TF binding prediction. And more specifically, we built our model on the architecture of this work as best set published in 2022, which is a convolutional neural network that predicts single-cell accessibility from the sequence. So in our work, with a similar, guided by a similar framework, we factorize the accessibility, the peak by cell matrix, into cell factors and peak factors. The peak factors are again encoded from DNA sequence, whereas the cell factors are encoded from single cell TF expression. And to understand this model, so first, the model captures biologically meaningful patterns in latent dimensions. So if we look at the cell factors and the peak factors and further reduce the dimension, we can see that the cell factors capture the rough trajectory structure in this mouse brain data set, whereas the peak factors moderately group together sequences with important motifs. So for instance, SOX11 and Neuro2 over here. And in general, on both held out peaks and held out cells, the model accurately predicts the TF expression and accessibility. And then to correspond, to extract information from this model to correspond the sequence information and the expression site, we take gradient of accessibility with respect to the sequence input and the single cell RNA expression input. And so we can see that from the sequence input, it captures the gradient times input of the sequence site, captures the important motifs. So for instance, we show this Neuro2 motif over here. Whereas on the expression side, we call this grad TF. It captures high correlation TF peak expression pairs. And I guess this figure is not showing, but essentially in the middle, we are able to find that it's more sensitive to low expression TFs. So we have an example of RunX1 over here. For some reason, that's not showing. And so again, to predict the TF binding, which the ground truth we compile from bulk ChIP-seq data sets, we compare it with a baseline logistic regression with motif matching score and correlation between TF expression and peak accessibility. And then we further include our two model metrics, and we find that it improves the prediction by a large amount and is comparable to another state-of-the-art method, Linger, which uses external bulk data in this PBMC dataset. And when we calculate the metric importance of these four features, so in general, we find gradient with respect to sequence to be the most important factor. And to further improve the prediction, we replace this single value of gradient sequence with this factor, with the peak factors. And this again further improves the prediction. So for example, we show four ground truth, four improvements over here. So in summary, our model, the gradient-based metrics, we call this grad-seq and grad-TF, improve the TF binding prediction. And this gradient with respect to sequence is the most important feature. And finally, I'd like to thank my lab mates and my supervisor.
Good. Thank you.
Thank you.